Hello, welcome back. Today we're going to look at what really drives, in terms of energetics, the synthesis of ATP in mitochondrion, where most of our cellular ATPs are synthesized. But keep in mind one thing, that's not the only place where cells can generate ATP. There is something else, also known as substrate level phosphorylation. And in fact, uh, prior to advocation of chemiosmotic theory by Peter Mitchell, although mitochondria was known to be the site of ATP synthesis, the mechanism of ATP synthesis was not very well known. And with the kind of data that they had access to, Peter Mitchell was able to put forward the chemiosmotic theory for ATP synthesis, and that fetched him a Nobel Prize. We're not going to go in great details of all these things, but we're going to only look at from the point of view of bioenergetics as to how cells harness energy and the mechanism from which the energy is harnessed for synthesis of ATP in mitochondrion. Keep in mind, when I say synthesis of ATP, it's not really de novo synthesis of ATP, it's just phosphorylation of ADP by ATP synthase, or F0 F1 ATP synthase, which is a transmembrane complex which is located on the inner mitochondrial membrane. Again, we're not going to go into intricate details of the respiratory complexes. Each one of them have got their own story, fascinating story. We're just looking at bioenergetics of it. Okay, I'm going to make a statement, and I'm going to try and make sense out of that statement over the next few minutes. The energy required for synthesis of ATP in mitochondrion is obtained from the free energy change of movement of electrons from low redox potential substances to high redox potential substances. And in this regard, I would really want you to be aware of a couple of things. One thing is what we typically refer to as free energy, okay, which is depicted by the term, by the symbol G, stands for Gibbs free energy, although there is Helmholtz free energy as well. Gibbs free energy is simply the maximum potential available in a system for doing reversible work, or in simple terms, it's the potential available for doing work at constant temperature and pressure. And when you look at the Gibbs free energy of the product versus reactant, you also determine what is called as delta G value for a reaction. And you've got to know that delta G values can either be negative or positive or close to zero. If delta G values for a reaction are at or close to zero, then the reaction tends to be at equilibrium. It reaches equilibrium in the sense that the reactions will be reversible. If the delta G values are negative, then the reactions are highly spontaneous. And if the delta G values are positive, then the reactions are spontaneous in the reverse direction, or they may require energy input. And this is something you really got to keep in mind. And another thing I want to be aware of is something called as redox potential. Okay, redox potential. Very, very important. If you understand redox potential, which is, by the way, very fancy, it's a fancy name for something that puts a number on the ability of any substance to either accept or to donate an electron. It's as simple as that. Okay, so we're going to look at all these things very quickly without actually going into intricate details of the working mechanisms of the respiratory complexes. If you would like me to make another video, let me know. I'll do another video later on. But let's just look at the energetics of it. Okay, now I'm sure you're all aware of this diagram. Okay, mitochondrial electron transport system has got complexes embedded within the inner mitochondrial membrane. We have complex 1, which oxidizes NADH, complex 2, which oxidizes succinate, complex 3, which reduces cytochrome C, and complex 4, which oxidizes cytochrome C as it reduces oxygen to water. Okay, and as these reactions happen, complex 1, 3, and 4 pump protons into the intermembrane space, okay, from the mitochondrial matrix. And because of that, the mitochondrial matrix, which is the space within the inner mitochondrial membrane or inside of the inner mitochondrial membrane, tends to be ever so slightly more alkaline than the pH of the intermembrane space, which is the space between outer mitochondrial membrane and the inner mitochondrial membrane. And this really creates a proton motive force, which is harnessed by F0 F1 ATPase as it synthesizes ATP from ADP. Okay, I'm not going to go into all these details, although I'll still tell you that when the electrons are taken from NADH, two of them are passed on to ubiquinone. Ubiquinone then passes the electrons to cytochrome C via complex 3, and complex 4 will take electron from cytochrome C and reduce oxygen to water. Okay, the story is same, except that when the reaction start with complex 2, the source of electron is succinate. From succinate, the electrons go to internal FAD, and then the electrons go to ubiquinone, and then electrons go to cytochrome C, and then electrons eventually wind up on oxygen as oxygen becomes water. Okay, so that's the story. But as these things happen, protons are pumped from matrix to the intermembrane space by complex 1, 3, and 4, and that's what creates the electrochemical gradient in the form of proton gradient, which is accumulating in the intermembrane space. And F0 F1 ATPase is a fascinating molecule. It's a large complex, has got a membrane bound F0 particle, which is a proton channel, and a stalk with which F1 particle is attached. F1 is the catalytic activity which carries out synthesis of ATP. And if you want to sever the connection between F0 and F1 particle, then F1 will behave as an ATPase. So you might have read in some books saying that it's an ATPase, mitochondrial ATP synthase or ATPase, both are same. It's just that the fact that energy required for synthesis of ATP from ADP is harnessed by the F0 particle as it allows protons to flow back. And the whole game lies in the fact that creation of proton gradient requires energy, and therefore by the law of thermodynamics, which states that energy can neither be created nor be destroyed, it can be transformed, it can be converted from one form to another form. When the creation of proton gradient requires energy, dissipation of it must yield energy, and that's what is going to be used by the F1 particle of F0 F1 ATPase to synthesize ATP. Okay, that's the story. I'm not going to go into intricate details of these complexes, that's for another day, but I'd like you to look at it from the point of view of bioenergetics, which we will do now. See, again, I'm not going to go for the sake of time, I'm not really going to go in detail about this equation, although very, very important. It tells you what's the free energy change for movement of protons from from matrix. Okay, this is the outer mitochondrial membrane, this is the inner mitochondrial membrane. Inside that you have matrix, and there are complexes here 1, 3, and 4. Okay, I'm not showing 2 because it's not participating in proton gradient formation. So all these pump proton and into intermembrane space. Okay, what is the energy requirement for this? Because it's an energy consuming process, it's not a spontaneous process. The energy requirement for this is about 22 kilojoules per mole. If you want to move a mole of protons across the mitochondrial inner membrane into the intermembrane space, you need about 22 kilojoules per mole of energy. That's very, very important. Okay, and in this equation there is something called as transmembrane potential. Okay, it's a very, very important concept. When the transmembrane potential is impaired, mitochondria tend to initiate apoptosis, and that sits as the underlying pathology of many degenerative conditions. And again, I'm not going to go into that. There are experimental methods to study that. If you want me to talk about it, let me know, we'll make another video. But where does this energy come from? Okay, see, in order to pump protons from matrix into intermembrane space, you need energy. And who's going to give you that energy? And that the answer to that lies in the redox potential of substances which participate in electron transport chain. Now, if you look at this diagram, okay, there's an equation that says that the free energy change of any redox reaction is this one. Okay, delta G naught prime, okay, is minus NF delta E naught prime. Okay, N stands for the number of electrons that participate in electron transport reactions, mostly 2. Okay, unless it's cytochrome C, because cytochrome C is a single electron carrier. But when you're looking at NADH as the source of electron, or FADH2 as the source of electron, or even prior to that, succinate, okay? Any of those, all of them can give 2 electrons. So mostly what you will plug in here is 2 electrons. F is the Faraday's constant, whose value you don't have to remember, but still a positive value. And this is more important, okay? Delta E naught prime, okay? It's just change in the standard reduction potential, okay? Or redox potential, whatever, right? It's simply the redox potential of electron acceptor minus the redox potential of electron donor. And unless you're involved in determination of redox potentials, don't worry about it. Just understand the values that are given. Look at the values given in textbooks and try to understand the concept as such. But redox potential values can be determined by electrochemical experiments, and we really don't have to worry about it. If you're a biochemist and if you're trying to understand how mitochondria harnesses energy for synthesis of ATP, all I want to just appreciate is redox potential of any substance. Let's say we have a substance X, okay? Now, redox potential of this substance would be a number, okay? It can be either minus or positive, doesn't matter. It's a number that indicates whether this has an inherent ability to donate electron or to accept electron. It's simple as that, okay? It's as simple as that. If it's got an inherent ability to donate electrons, then the reduction potential values will be low. That's what you got to keep in mind. And if it's got an inherent ability to accept electrons from someone else, its reduction potential values will be very high comparatively, okay? And the low means it can even be on the negative side. Obviously, high means it's on the positive side, okay? Not necessarily, but on. See, when you're looking at a redox reaction, it's always a pair of substances that participate in it. That means if there is a molecule called A, which got an inherent ability to give electron, it has to come in contact with somebody like B, who's got an ability to accept electrons, okay? It's as simple as that. So if the redox potential value of B is higher than A, then invariably electrons will move from A to B. It's as simple as that. And when that happens, if you plug in here these values, you will see that for the movement of electrons from NADH, and where does NADH come from? It comes from all the catabolic pathways like tricarboxylic acid cycle or beta oxidation that's happening in mitochondria, or even other enzyme or other even metabolic reactions that are happening in cytoplasm. But in order to move cytoplasmic electrons which are present in NADH, you need participation of what is called as shunt pathways. Mostly you can look at what's called as aspartate managed shuttle, and you'll understand how electrons move from cytoplasm into mitochondria to be oxidized by electron transport chain. Which will go into reduction of oxygen. But NADH, FADH2, all these are the currencies that you generate, intermediate currencies that you generate in the catabolic reactions, which are then going to link catabolic reactions to electron transport chain for synthesis of ATP. And if you plug in the values for electron acceptor and electron donor, then you will see that the delta G values will be negative. It's as simple as that, okay? And that means that these are spontaneous reactions where the energy of the reactant is more than the energy of the product. So the difference is always going to be released out. That means it's available for doing some work, and it's exactly that's what used up by complex 1 and 3 and 4 to pump protons, okay? So we'll take some example of that and we'll try and understand, okay? Let's move on to the next slide, okay? Now what you're looking at here is the reaction where you are involving complex 1, complex 3, and complex 4, okay? So complex 1 is the one that oxidizes NADH, and NADH has got a redox potential of minus 0.3, and eventually from complex 1 to complex 3 to complex 4, eventually to oxygen, electrons will be accepted by oxygen, whose redox potential value is about 0.8. Now, if you plug in those values, you will see that the delta E value, which is the difference in the redox potential of the acceptor and the donor, you will see that that value is about point whatever, right? You're plugging in these values and you say that comes out to be a positive value, okay? 1.14 volts. And if you plug that into this Nernst equation, which is delta G naught prime is equal to minus NF delta E naught prime, 2 electrons are moving. This is the Faraday's constant, and you're plugging in delta E naught prime values, you will get minus 220 kilojoules per mole, okay? Of when you oxidize a mole of NADH, you're going to get minus 220 kilojoules per mole. And it is this energy that is harnessed by complex 1, 3, and 4 collectively to pump protons into intermembrane space, thus creating proton motive force, which is going to be reversed by F0 F1 ATPase as it synthesizes ATP. It's as simple as that. Okay. Now, what I have done is I've listed here the delta E naught values for individual complexes. Just have a look at it. Complex 1 has got a delta G naught value of minus 69 kilojoules per mole. Complex 3 has got delta G naught value of about minus 37 kilojoules per mole. Complex 4 has got a delta G naught value of about minus 112 kilojoules per mole, which is far greater than 22 kilojoules per mole, which is required for pumping protons into intermembrane space. So all these three complexes energetically are sufficiently good enough to pump protons, and therefore they pump protons into intermembrane space. But complex 2, which catalyzes oxidation of succinate to fumarate, has got delta G naught value of about minus 16 kilojoules per mole, which is less than what is required, which is 22 kilojoules per mole, which is what is required for pumping protons into intermembrane space. And therefore, complex 2 energetically is incapable of pumping protons into intermembrane space. Okay, so this is what is the story of electron transport system by using the energy change, free energy change that is associated with movement of electrons from low redox potential substances to high redox potential substances. Cells harness energy there and they couple it to creation of proton gradient. Okay, and that proton gradient is dissipated back into the matrix by F0 F1 ATPase, and thus the energy is now available for synthesizing ATP. Okay, so complex 1, complex 3, and complex 4, whose delta G naught prime values, which are greater than minus 22 which are greater than 22 kilojoules per mole on negative scale, but it simply means that these spontaneous reactions will give you enough energy output to meet the energy requirement for pumping of protons into intermembrane space. Thus, you're creating a proton bank or transmembrane potential for proton. And transmembrane potential is one of the greatest driver of a lot of biochemical processes, okay? Whether it is neurotransmission or whatever, right? So once transmembrane potential is created here in the form of proton gradient, it's going to be dissipated back by ATP synthase to synthesize ATP. It's as simple as that. Not at all complicated, okay? So I hope you have fairly good understanding of how energetically synthesis of ATP is allowed in mitochondrial situation. If you'd like me to talk about these complexes individually, do let me know. I'll be happy to talk about all these complexes 1, 2, 3, 4, and 5 because they've got fascinating stories of their own. Complex 1 inhibition recapitulates pathology of Parkinson. Complex 2 inhibition recapitulates pathology of Huntington's disease. Complex 3 is a very fascinating, redox complex where there is something called a skew cycle, which shifts gear from 2 electron transport system to 1 electron transport system. And complex 4, we all know what complex 4's importance is. If you choke complex 4, everything is going to shut down, and that is the target, one of the targets for cyanide ions. Okay, and complex 5 itself, which is F0 F1 ATPase, has got a very fascinating story. Okay, it can synthesize ATP, it can hydrolyze ATP as well. It's just that under cellular circumstances, ATP synthesis is allowed because it is energetically permitted or favored by the proton gradient. Okay, and keep in mind the proton gradient which is created here in the intermembrane space not only drives ATP synthesis, but also it helps in movement of ATP out into the cytoplasm and taking cytoplasmic ATP back into mitochondria for rephosphorylation. So that exchange is also favored by proton gradient. Proton gradient is critically required for importing proteins into mitochondrial matrix. And once the proton gradient is lost, or transmembrane potential is lost for whatever reason, mitochondria can trigger apoptosis, and that's the importance of proton gradient. And now I hope through this video you understood how energetically mitochondria are in a position to create proton gradient because of movement of electrons from low redox potential substance to high redox potential substance. I hope you'll enjoy this video. If you enjoyed it, please subscribe to my channel. It will only encourage me to post more and more videos. Thank you so much.